Okay, just to finish up the cell lecture for you so you feel like you got everything out of that posted outline, we left off talking about genetic diseases, and the idea of a genetic disease is that because the mutation is in the germline cells, that's either the egg cell or the sperm cell, then once that mutation shows up in an offspring, it shows up in all the cells of the offspring carrying the mutation. So assuming it's a non-fatal mutation, it's going to exist not only in that individual but also passed down through that individual's own offspring. A good example of this is sickle cell anemia. And in sickle cell, we know the defect is in the gene for one of the two polypeptides that hemoglobin is composed of. We also have treatments for this. Those treatments, though, are for that individual, and as yet we have no genetic engineering solution to prevent that trait from being passed on. I also want to point out that not all mutations are bad. As we saw in class, some of the mutations, the substitutions may be neutral simply because the codon of the RNA may change from one codon for another codon of the exact same amino acid. So they're like invisible mutations, and that kind of mutation can accumulate in the genome, and that's the basis of things like DNA fingerprinting, when we can tell your genes from the genes of anyone else, because everybody has their own individual mutations. Some mutations are in fact beneficial. One of the things I've read about most recently is a mutation in the gene for the muscle protein, and there are like 4 little kids around the world that have really strong muscles. Their muscles are real defined even though they're like 4-year-olds, and it's because of a mutation in the gene for one of the muscle proteins. So your genetic inheritance is based on your DNA, and this web length will take you to all of the animations that are available. By the way, in the PowerPoint for the outline, all these links are live. Some of these links are going to be bad links because they go to files that I had on my desktop, but here you can see the actual web address. So all of the animations I showed in class today can be found at this Biointeractive website, and I would encourage you just to go explore if you have time. I do also want to point out that mitochondria have their own genes. There aren't very many genes in mitochondrial genome. I think it's between somewhere around 8 to 25, and they're almost all related exactly to the needs of the mitochondria, but sometimes those are mutated, and those mitochondrial gene mutations can lead to genetic disease. In fact, recently in England, there was a new ordinance passed that allowed the experimental creation of a fertilized egg that had the mother who donated the egg nucleus, the father who donated the sperm, and then a second mother who donated just the mitochondria. So technically this individual, if the pregnancy persists successfully, will have 3 parents, because mitochondria generally come from the egg, that is, they're passed down by the mother. And another thing I want to point out is that the realm of epigenetics is something we've only recently become aware of. DNA is regulated. Genes are turned off and turned on by chemically adding or removing little organic groups to segments of the DNA. And those changes to how the DNA is regulated can be passed on from parent to offspring through, we know, at least 3 generations. So at the heart of it, there have to be genes involved in all that regulation, but it's interesting to note that environmental changes that affect an individual can affect how the genes are expressed in his grandchildren. Okay, so we talked about the Leber disease, LCA, and we looked at the changes in the retina. I'm going to go through these very quickly because they were also in the other recorded PowerPoint. And the effect of the ornithine cycle, that enzyme that is deficient and causes ammonia buildup. And the idea that genetic engineering can be used to actually fix genetic mutations. And we talked a little bit about the cell cycle, and one of the things that we skipped over this slide, I'm not really sure why, but we did talk about the gap 1 or gap 0. We talked about the S phase. We talked about the gap 2 phase. We didn't focus in that much on the phases of mitosis, and I do want you to be familiar with them, but I don't want that to be the sole focus of your study of cell division. Mitosis is the changes that we can see under the microscope as the chromosomes are prepared for division. And it is followed by cytokinesis, which is the actual pinching in half of the two daughter cells. So the nuclear division, that is the division of the replicated chromosomes, that's mitosis. And cytokinesis is the division of the entire rest of the cells. I did mention that you need centrioles and microtubules. In fact, cells that lose their centrioles cannot divide and can't replace their centrioles unless they have the base of a centriole, which happens to be the same as the base of a cilia, which is an interesting problem for another day. And I wish we had had time to get to this. I alluded to it briefly, that if a cell loses its ability to regenerate, then aging can be accelerated, and aging or senescence is a loss of the cell's ability to divide. We know quite a bit about this. We unfortunately do not know what causes progeria, which is the apparent acceleration of aging in children. It's not truly aging, but it sure looks like it. And so you have these kids, 3, 4, 6, 12 years old, dying of age-related disorders like cardiovascular disease, heart attacks, showing male pattern baldness. They've never hit puberty. They're never going to. And yet this problem of aging is accelerated, and we know that it's because of the cell's inability to regenerate by going through mitosis and cytokinesis. So that certainly points up the importance of cell division in maintaining a healthy individual. And we did talk at length about cancer being kind of the opposite problem. So cells kind of walk a tightrope between not wanting to divide too much, because that would be out of control cell division in cancer, versus not being able to divide at all, in which case the cells age and die. So remember that with each division, the fact that the DNA is being replicated makes it more susceptible to mutation, and so the more often cells divide, the more likely they are to mutate. So cells that go through repeated division actually accumulate mutations up until the point where they undergo a fatal mutation and the cell dies off. So chemotherapy and radiation therapy both attack the Achilles heel of cancer cells, which is that they're rapidly dividing cells. So we know that the DNA is replicated in the S phase. We know that it's more vulnerable to mutation then because it splits into its two sides, two helices, and that each side is replicating the other side, which makes it unable to repair properly. Radiation, we remember, pops out the nucleotides, breaks the chemical bonds between the nucleotides, and so the rapidly dividing cells are the ones that go through the cell cycle more frequently, which means they're in the S phase more often. All right, and don't forget you have an assignment for next week. I want you to become familiar with how did you get to the point of fusion of an egg and sperm, where did those chromosomes come from, and the formation of ectoderm, mesoderm, and endoderm. See you Monday.